So this is the overarching theme I'm going to talk about. It's protein structure, function, and catalysis. And enzymes. They are molecular machines. And you can look at them like this, like we quite often see them. But today, I'm not going towards the complexity of networks. I'm going to look at the complexity at the atomic scale when they work as machines, and what they do, how they do work at the atomic scale. So thanks to Phil for a very nice introduction to the field of cellulose deconstruction. This is the substrate we're going to look at today. It's chitin. It's very similar to cellulose. And what happens during catalysis is that these gray hydrogen atoms here is pulled off, and then this chain is cleaved afterwards, after the enzyme is finished. The enzyme was discovered in 2010, and it was thought to use molecular oxygen as a co-substrate to cleave and oxidize these polysaccharide chains. Last year, however, it was found out at NMBU that it's not oxygen, it's actually hydrogen peroxide that is used to replace this hydrogen atom with a hydroxyl group. This is what the enzyme do. But how does it work? This has been a topic with huge controversy. And I will address two questions today. How does the enzyme interact, bind, and orient itself on the substrate? And the second question is, how does it perform catalysis? What does the energy landscape look like here? And first, how do the enzyme bind to the substrate? So this is the surface of a crystalline polysaccharide. And this is the catalytic core inside the enzyme. And it has to come close to in the correct orientation and position to perform catalysis. How do it do that? You can start guessing, but it's quite enormous amounts of orientations that you have to explore to figure this out. And it's not feasible. So, help from experiment. We look at cutting patterns, and we find out how the enzyme binds to the substrate in a particular orientation. We use MD simulations to look at how the surface interacts with the polysaccharide chains. And we see that there's a conserved patch of amino acids that is mainly binding to the substrate, actually. And how does the co-substrate, hydrogen peroxide, get into the active site? When we look at the enzyme on the substrate, we see an access tunnel. And we see that it's gated by amino acids. And we find that these hold specific orientations. And by looking at models, this is a kind of a 5-minute computer laptop calculation. We can look at how these possible ways into the active site from outside to the copper site can be found in space. But you can also look at this and look at what kind of energy it takes, what resistance the hydrogen peroxide takes to go into the active site. So this is the potential energy landscape. And now we see here we are pulling the hydrogen peroxide towards the copper here. We are now trying to jump over this hill. And now it's slipping through and getting close to the copper atom in the active site. So this is more like a week using hundreds of computers calculation to figure this out. It gives us an idea how the active site is restricted and how substrate is gated into the active site. So, what happens next? The hydrogen peroxide is in the cavity close to the copper. The enzyme is bound to the substrate itself. Here is the model. This is the enzyme part, hydrogen peroxide, substrate. This is the bond to be cleaved. This is a quantum mechanical simulation, so we can look at bond breaking and formation. It's the hydrogen peroxide. So first thing is happening is that the hydrogen peroxide is split into two. This generates a very reactive hydroxyl radical here, which next snaps off a hydrogen here and generate a very highly reactive copper oxygen species, which is now ready to take off this hydrogen. It does. And now this oxygen recombines to this carbon to form a new bond. And this is the final enzymatic product. So this is a movie showing how it goes on. Is it feasible? We have to look at the energy landscape of the reaction to figure out which steps are the most energy-consuming ones. And here we can see the final energy landscape of the mechanism. We have probed the bottoms and the hills of the energy landscape, and we see that the splitting of hydrogen peroxide itself seems to require quite a lot of energy. This peak here, this is the hydrogen abstraction itself. Requires almost no energy compared to what it does without an enzyme. This bond is very, very difficult to break. So here we see and understand how the enzyme can break these tough bonds in the polysaccharide. So the main implications here, we understand, we get the beginning of understanding how LPMOs interact with substrates. We're going to look at this for more LPMOs, more substrates. But we've also found that this active site channel may also play a role that we didn't know of before, restricting the fusion of reacting molecules into the active site. And how we can explore this information, knowing the reaction mechanism in detail, is important when we want to mimic these biological systems and the chemistry they are carrying out. So that's it. Thank you very much. Any questions?
I'm supposed to ask that question, but go ahead. Yes. Any questions for Åsmund? We have 2 minutes. That was apparently very clear.
Yes. In the living environment, it's probably generated by oxidases. There are many enzymes that use oxygen as a substrate and then oxidize something else and generates hydrogen peroxide.
Okay, thank you again, Åsmund. Here's a little present from the organizers.
Thank you very much.